All right, so today I'll talk about perception of color and the computation that underlie the perception of color. It's going to be a very visual lecture because so much of color involves us actually seeing color. We're very responsive to color. So let me briefly make a few introductory comments to vision. So vision in general, particular vision for us primates, for humans and monkeys that have high resolution image sensors, particularly at the fovea. So image, we can at a single glance identify a lot of things from the world just by using this distance sense. It's a sense that acts remotely, unlike a touch or smell that typically involve interacting with objects close by. So just by looking, in this case, is one of the most famous paintings ever done. You can identify the gender, you can identify something about her state of mind, you can identify whether she's looking at you or not, whether she's upset or not. This is a beautiful, one of my favorite painter by Canaletto. There's an enormous amount of cues hidden in this image. Of course, it's a two-dimensional image, but we can all very quickly infer the three-dimensional relationship of these objects by a whole variety of cues, including perspective cues. Right, so here you can see very noticeable this was only discovered 150 years prior to this in the late Renaissance, when people discovered that you can use perspective cues to give that gives rise to very powerful depth perception. But there's also other cues as shading. Things in the background tend to be more bluish. It's the size of people that we're accustomed to that gives us something about the depth and the range of depths in this image. We also can use our vision to alert us to potential dangers. That's of course one of the key functions of this smell. We can use it to infer, even from stationary images, we can use it to infer motion in this case. And even when we remove a lot of the high spatial frequency detail, like Turner did, you can infer a lot about the spatial arrangement. You can all see this is interior room, and you can sort of see the sun coming through and the walls and the furniture in front. And particularly, you can infer a lot about the color. Right? All these paintings are colored. So ultimately, if you think about what vision involves in visual perception, the best way to think about it, the way I think about it, is to paraphrase what David Maus, a famous theoretician of vision, said, that visual perception is really the construction of a description of the world. So we look at this painting, early surrealist, de Chirico, and we infer something about the world, and we construct very rapidly, within a couple of 100 milliseconds, we can construct a description of the world. In terms of what? So what is there? In this case, a torso and bananas, object recognition. We can infer something about the surface property by looking at them, partly just purely from vision, partly because we have a rich association with torsos and with bananas, and we have certain expectation about the surface property. In particular, I'd like to point out now the color, because that's what we're going to talk about today. So you have all of this object that has sort of basically one sort of color, and these objects have a different color. And so the way to think about color is really an attribute of surfaces. Classification of action, like less here, but in the previous matrix reloaded, you can see those Agent Smith and Agent Anderson, Mr. Anderson, they were fighting. And of course, you locate things about where, the spatial relationship among objects, amongst each other, the shape of objects, and the distance. And ego motion, again, not in this case. And so once again, if you think about perception as a construction of a description of the exterior world, theoretician in vision, both computer vision as well as biological vision, sort of distinguish three separate stages in vision. There's the early stage, there's a mid-level stage, and there's a high-level stage. We don't know to what extent these are all realized in mice. So early stage involves things that we do in the retina or very early on, where we inquire information about some of the features of the raw features that make up visual information. So for instance, changing velocities, changing image intensities that we interpret as motion or motion flow, the intensity of pixels in the world, their color, their texture, particularly the existence of edges. Right here you have these very sharp edges. In the Turner painting you had very few edges, but of course you can still recognize things. stereo. So it turns out that our vision is amazingly robust, and while we use all of these and more primitive features, we don't really need any one of them. Of course, typically we need one or two of them combined, but we can miss any one or two or three of them and still see things very well. So then the mid-level vision involves the specific computation of color that typically is considered a mid-level vision computation. We group things. We organize things into boundaries where we say, well, all of these edges correspond to a single boundary, the boundary of a person. We do figure ground. It's a very powerful computation where we rapidly say, well, this is all a figure, and then there's the background. And then finally what we do, and there are two distinct pathways that have been quite well researched in humans using patient and psychophysics. There are really two sort of outputs for this visual process. And once again, we know much less about this. In this, we know very little in mice. There is the vision to motor, right? Because ultimately I have to act in the world. I have to run, I have to bike, I have to reach out, I have to eat, I have to interact with people. So all that requires that I recognize where objects are, how they look, how they're shaped. I have to reach out and grab things, so I have to know what the orientation is. So that's for motor. But then we also do all sorts of cognitive computations with vision, right? We read text, for instance. We identify features of faces of the people we interact with. It's a very important. And if you have deficits in that, like if you're face blind, you're significantly impaired in your social interaction. So we really have two outputs. Mice are also very likely to have both, but we know very little about, in particular, about this component. And we hope in the next 10 years that we learn a lot more about visual cognition in mice, doing both behavior and electrophysiology. So we're going to focus on color, which really is people used to think color is sort of really already out there in the world, but it's really much more something that has to be computed explicitly. It's not, see, stereo and motion, there's a physical variable that corresponds to stereo. So, if Hongkui looks at me, there's a physical distance between her eyes and my body. There's an objective physical variable. If I move, Hongkui's visual brain can extract how many, the extent of motion. Again, that's a physical variable. Color really isn't like that. Color is really quite different. Color is really something that's there's a continuous wavelength, I'll show you, and the brain abstracts this and assigns a particular label to it, and we call this label color. So this involves a domain between early vision and intermediate vision. And in the visual system, color is extracted and computed in various stages, from the retina to the LGN to primary visual cortex to higher order visual cortices. So I like to talk about color because it really teaches us a lot about our minds. We all believe instinctually and universally that objects look colored because they're colored. Right? An object is obviously colored. But that's actually not the truth. Color is an attribute that we assign to surfaces. Many people assign different attributes to the same surface, and many species will assign different colors to the same surfaces. So it's not a universal physical variable as is depth or as is motion. It doesn't correspond to any simple physical property. Of course, it relates to physical properties, but it doesn't correspond to any simple physical properties. It makes it very interesting from a computational point of view. So first of all, color matters. So here you see a scene of mountains. This is, I think, outside Woods Hole, a lake and some trees, and here you can see them with color. Now, of course, you immediately extract much more information. You can see the state of the foliage here and how advanced they are in foliage. And in fact, there's belief that the evolution, particularly of trichromacy, what most of us have, where we have three photopigments, partly is driven by sort of foliage and fruits, the ability and the need to recognize the state of fruits and foliage in forest. Here's another picture. You can observe these, you can't really see these, the flowers here very well, but here they instantaneously pop out. The colors matters a lot. Although it hasn't been that easy to establish that quantitatively. This is from a short Nature paper where actually they show that color really matters for a sports event. So here they went to the 2004 Olympics and they looked at the way these are all fighting sports, combat sports, and what the two parties wore, whether they had blue, well, it was red versus red versus non-red, I believe. And here you can see, so here they average over all these sports, boxing, taekwondo, Greek-Roman wrestling, and freestyle wrestling. And you can see the more, particularly here, the more people are balanced, the less. So if there's a large degree of symmetry between wrestlers, for example, in terms of their ability, in terms of their size, then color doesn't seem to matter a lot. But if there are no objective criteria difference between their rankings or their weight, then color, then if you have red, you get this hugely enhanced significant boost. It's quite striking. We still don't know whether that's a purely perceptual effect or it's a hormonal effect, but it's very interesting if you wear red. And of course, if you go into Victoria's Secret or stores like that, what you'll see that lingerie by and large all tends to be red colored. Now, all of that may possibly relate to the side of blood. Blood, of course, being red, venous blood, arterial blood is red, and so the question whether both the sex as well as this violence effect relates to that right now, that's a question of ongoing research. It's very interesting. People tend to buy less blue lingerie than red. So the modern study of color starts, like many things in science, with possibly the greatest scientist to ever live, Isaac Newton, who as a young man already experimented with optics in his in college. And then he wrote this book, Optics, in I think 1704, something like that. That's not quite as well known as his Principia, his treatise on gravity. This is sort of more empirical based, while in the Principia, he talks a lot about theory, and he talks, of course, about what he called fluxons, which today we recognize as differential calculus. Here he talks about more experiments he did as a young man. And amongst other, he first coined the term spectrum, which sort of typically means a spectral appearance, right? A ghost-like appearance, because one of the things he did, he constructed a spectrum, although surely this is done probably previously by other people, but he was the first who wrote upon it. You take a piece of glass and you take sunlight and then you split it up into these various components, and then of course you can recombine them using another spectrum. You can recombine them again into pure white light. And so he had this, this was one of his underlying theoretical assumptions, that there is a certain power and disposition. Today we call it the wavelengths, or we call it the energy of the photons. Sort of he had this idea roughly 300 years ago. That's how colors are part of. You have a white beam and you can split it up into the individual components. So this is today what we call a spectrum after his term. So this is the entire electromagnetic spectrum. And of course, we see only in the visible light. Now, we humans typically only see from here till here. Mice, they can see all the way down to here to 350 nanometer. I'll talk about that later. They can see in the near UV. We can't. And of course, some animals like rattlesnakes, etc., can see over here in the near infrared around 800, 900 nanometers. But sort of humans, we are restricted to this domain. Using our natural body, which is just, of course, a part of a much, much larger domain of all waves, including from gamma rays all the way down to shortwave radio. So it's less than a factor of 2 that we can see, right? So typically from 450 to 700 or so. So it's a very small domain. So if you think about everything we can see, and all these wonderful colors are all packed into that very small spectral band. So how is color generated? Well, so you have a light source. Evolutionary, of course, the only light source, except for the occasional fire, is the sun that radiates across the entire spectrum, right? The sun, of course, has gamma rays, although not that much like compared to other gamma stars, but it radiates across the entire spectrum. In particular, it radiates in the UV, and none of us would be around here if we wouldn't have ozone and other things in the atmosphere that absorbs a lot of that UV. So the sun radiates, or any light source like these artificial light sources, they'll radiate broadly, not just in one particular narrow spectral band. So here you can see this is a spectrum radiation of the sun, right? So evolutionary, this is really the only one that mattered. Typically, I mean, this is it corresponds to a physical thing called the black body radiation. with roughly a surface temperature of around 5,000 degrees Celsius. So this is what radiates out. So this is what you can actually measure outside the atmosphere. If you're a satellite, 50 kilometers out, that's what you measure. Now what you actually measure down here on the ground on a totally overhead-free day is sort of this red spectrum. You're missing things here because of the absorbing effect of the atmosphere. So these are water bands that take out this radiation. In particular here, what's really important for us, for all of our well-being, is the fact that O3 here, ozone, takes out a lot of this UV. This is pretty harmful for us. And of course, if you get a sunburn, that's because the action of the UV. And so if we would miss that, due to our nefarious activities in the atmosphere, then bad things will happen to us and to all sort of other light. Anyways, you can see it's a remarkable steep fall-off here. So this is actually what evolutionary we and all animals sort of evolved, this sort of spectrum. But once again, we see typically only between here, 400 or 4-something 20 for primates, all the way to over here. This is all invisible to us. All right, so that gets emitted in all the angles for the sun and in all the different wavelengths. So that's the illumination. And then it strikes an object like T-dogged. It's the dog of my German Shepherd of my daughter, Tosca. And then what you get reflectance. So some of this wavelengths as a function, some of this energy as a function of the wavelengths and as a function of the orientation of her surface. And of course, this is a very difficult surface as compared to metal surface where you get very simple optical reflection, because here you get her fur. It has very complicated reflectance property. But ultimately, any substance, whether it's grass or fur or ice or stone, will reflect a certain fraction into a specific angle, whether it's very directed like in metal or whether it's diffuse. That depends on all sorts of surface properties that people care a lot about, particularly in the computer graphics industry. Of course, they care a lot about that because they want to probably model objects like a German Shepherd dog. So you can see here, so this is 3 different properties: grass, soil, and water, and their reflectance properties as a function of the wavelengths. Going from UV 400 nanometers to 700 nanometers. So you get differential reflectance as a function of the object. But note always that in all of these cases, for example, water or soil, you do get wavelength reflectance across the entire spectrum, right? It's not that you just see, when people say red, well, it's not that you only see 700 nanometers or green is only 600. You always get these very broad reflectance across the entire spectrum. Okay, now that gets reflected all over the place, and some of that energy, depending on the orientation of the object, its surface relative to our eyes, will ultimately be absorbed by our photoreceptors, right? So the photons originates here in the light source, travel all the way through space until they hit an object here, and then some of them, a small fraction, of course, are reflected into our eyes. And there they get absorbed, and that's the source of the signal. And now we do a lot of processing, and then this is done by the brain, and then you become actually conscious of the color, and you use it to label objects. And so we want to understand how this happens. But here the important point I wanted to stress here, this step, there's not a single, I didn't talk about single colors here. I talk about these continuous reflectance function. And that get finally absorbed by 3 discrete classes of photoreceptors, at least in most of us. In some of us, particularly the women, it gets absorbed into 4. In some of the guys, it may only be absorbed into 2 different classes of photoreceptors. So it raises a question: how can spectral information, I mean wavelength-specific information, be used to identify and characterize different surfaces independent of the light source? So you see what you want, depending on the light source and depending on changes in the light source. For example, when the sun is low in the morning, right, it has to go through a whole lot more atmosphere, and it changes its light property than when it's high up in the sky or late at night. When it's setting, in both cases, it can changes from yellow to red. It has to do with the absorption properties of the atmosphere. Or when it's indirect light, like in the case of moon, right? Moonlight, of course, is just indirected it's just indirect reflected sunlight, so that looks very different. But we want the surface, and our brain does this partially, it's called color constancy, that you can see this jacket always as blue, even though it might be moonlight, or it's this artificial fluorescent light, or it's sunlight. And if you put a photometer out and track me around, I can assure you that over a 24-hour period, you'll get a very different spectrum reflectance function. You'll get a very different spectrum inside of the photometer if you measure it across the different wavelengths. But by and large, you will always tend to see this blue. It's not completely true. So people have what's called a weak color constancy. So color constancy refers to the fact that you see, that you perceive as the surface attribute known as color relatively constant, even though the light source might change, and therefore what the spectrum of what you get in your eyes changes. But your brain still tries to extract that underlying surface property independent of the nature of the light source. So the input to the retina is an incident wavelength-dependent light distribution across the spectrum, while the output, the one that you see and that we use to label and to do all sorts of computation, is a color percept that labels the surface in an illumination quasi-independent, not fully independent, but a quasi-independent manner. That's what happens with color. And there are really three principles of color perception that took scientists the last 250 years to work out, really the last 150 years to work out. There's the principle of trichromacy, that most of us will perceive, that we have three different color receptors, or put differently, that you can match any color of any natural object by just three colors, three primary colors. That's the principle of trichromacy. It's not universally true. As I said, it's not true for many women. It's not true for roughly 7% of guys. The principle of opponency, and these used to be things that were heavily fought over, and people used to think, well, either it's trichromacy or opponency. It turns out our brain, depending on what stage of the brain you look at, of the visual system you look, represents things in a trichromatic manner or in an opponency manner, and the principle of color constancy that I already alluded to. All right, let's talk about trichromacy. So this is known also as the Younghelm people. So most normally sighted humans, color vision can be expressed by three numbers. And today, that seems obvious to us because today, of course, we know that there are three different photoreceptors that we use during this sort of vision, during cone-dominated daylight vision. And they're conventionally labeled, but somewhat incorrectly labeled as red, green, and blue photoreceptors. They should really be labeled as, and typically they are in the scientific literature, as long wavelengths, medium wavelengths, and short wavelengths, photoreceptor L, M, and S. That's really the correct way of labeling it, because once again, there are no red photons or green photons or yellow photons. What that means operationally, that in normal trichromats, any perceived color can be matched by a combination of three different wavelengths. Now, think of it, it's an enormous reduction. You go from this continuous wavelength distribution that's incident into your eye to something that spans a three-dimensional space, the three-dimensional color space. And what this implies, the existence of metamers. Metamers is a psychological term, comes from the Greek, that implies that things can look alike but actually be different, that you have two different surfaces or two different light sources or two different objects that have different reflectance property, but they look exactly the same. And then they're called metaphors of each other. Even though they might be different, they look to you the same. And there are variants of trichromacy in all of us, in the human species at large. They're called tetrachromacy, dichromacy, monochromacy, or achromats. People who have no color whatsoever, they completely lack cone photoreceptors and they only have rods. So this is experimentally how you do color matching. So you look inside a box, and what you see is this split screen, and down here you have this split circle. So on the left you have some test light, in this case sort of purple, and then here you have three adjustable primary lights, and you adjust them, the red, green, and blue guns on these, if they're, for example, CRT or something, or computer screen, you can adjust them until you can match them. And the claim is that any natural light can always be matched by some combination of these three lights. So that's the operational way to define trichromacy. Now, for some people, it turns out all you need is two colors. They're impoverished, and we call them colorblind, although technically the vast majority is not colorblind. They're just impoverished in their color perception, and most of the time it's guys who have this. And there are women who have, because of vagaries of photoreceptor of X inactivation, they actually have 4 photopigments. There are 4 cones, 4 types of cones with 4 different photopigments. There are 2 types of red photopigments in the general population. So among men, roughly 50% will have 1 photopigment and 50% have a single point mutation in their opsins for the L photoreceptor, and they have I'll show you a curve that's shifted by 2 to 3 nanometers, and they'll see red slightly different. Now, if you have women that have both of these alleles expressed in their eyes, for instance, because they have 2 sons that each have the separate one, then they have 4 different wavelength receptors. What that means, and it's been difficult to test, because the difference would be very subtle, that in principle, those women, they would need 4 lights to match any particular surface. That 3 lights wouldn't be sufficient, that they need 4 lights, that they could always tell the difference, although it might be minute. And so that's something that's currently being investigated.
And this is all presumably because the red and green cones photopigments are on the X chromosome.
Correct, yeah, it's sex-linked. Yeah, I should have mentioned that. All right, so here, so it's once again to return to what a metamer is. So here, if you have your 2 different, let's say you have your M cone, your medium wavelength cone, and your L cone, your long wavelength cone. So this is sometimes called the red, and this is sometimes called the green. And let's say you have 2 light beams here. One of this is green intensity, and this is red intensity. You can always match that by a single one that looks yellow. That is yellow, because if you adjust the brightness correctly, you cannot distinguish red and green from yellow. They look the same, although physically they're different. I mean, they're different photons here with different energy, but the combination of this will look like that. So that would be a metamer. And there are lots of other metamers. So this is, for example, from Mondell's textbook. You can have these 3 different distributions, and of course this changes from person to person because it depends on all sorts of things. Particularly, it depends on the exact distribution of cone photoreceptors in your eyes. To really make a metaphor, but in principle you can match these 3 very different looking. Okay, this obviously is from a technical device, right, that emits very narrow bands. This I think is fluorescent, and I don't know what this is. And you can match these so they all look the same. In fact, that's how TV works or movies work, right? Because they have very narrow the screens on our monitors, yet they give rise to the whole gamut of naturally occurring colors. That's because of metamers. So as I mentioned, throughout the 19th century and well into the early part of the 20th century, there were these debates among psychologists and physiologists whether the brain represents color with 3 colors, that's the Younghelm theory, or whether it represents in an opponency manner. This was one of the German guy, psychologist, Ewald Hering, who postulated that colors are essentially expressed in your mind and in your brain by opponency principles. In particular, he postulated 3 axes. We all know in normals it's a 3-dimensional space, and there's the bright-dark opponency, then there's a red-green opponency, and there's a blue-yellow opponency. And part of the reason was, I'll show you after effects, the other reason, and the argument was that there are certain colors that you can never observe simultaneously at the same location. You can only observe one or the other. So this would be in the color space. So if one axis for the intensity that vary between the poles of bright and dark, and then the other one blue-yellow, and this one red-green. And of course, that's also in our brain. I'll show you. In fact, Clay did some early recordings. I'll show you from his lab, where you can directly see a manifestation of this opponency principle at the level of single nerve cells. So this, you probably have all seen this in school, these sort of color after images. If you just fixate your eyes, put them here at the center and keep them still for as long as you can, let's say for half a minute. Don't move them. These things work best. These after effect works best because they're location dependent. If you really can keep your eyes very stationary, fixed on that spot. And keep your eyes fixed. And you all see the American flag? It's quite powerful. And it's very interesting. If you do this again, if you look now the second again, just fixate, keep your eyes still, you see A, it takes some time for the after image to be created. And then B, it waxes and wanes. That's in ways we still don't fully understand. So first you see a strong after effect, then goes away, it comes back, weaken, goes away. Sometimes you can see up to 3 of these waves appearing and disappearing. And now I'll show you my favorite all-time best ever top on my top 10 list of visual illusions, the lilac chaser. This is beautiful. It's awesome because after a while you see what's not there and you don't see what's there. So just fixate the cross here. Don't move your eyes. And tell me what you see. It's quite a new discovery. So if you just sort of move your eyes over it, what you see are 11 purple disks, right? There's one always missing, and this travels, the whole is traveling around. And after a while, you will not see the existing disk anymore. You will only see the whole, but in fact, you'll see the whole as an after image, as green, right? The after image to this sort of magenta is green. So after a while, and most of you should see that, if you keep your eyes steady, the center, you'll only see a single disk, right? Now, the single disk doesn't exist. The color doesn't exist either. It's all a construct of your brain. The only thing that exists are 11 magenta balls that you don't see. It's amazing, our visual system. I mean, it's amazing that you can navigate through the world. Who will build something like this? So it's a consequence. There are 3 different illusions in here. There's apparent motion illusion. There's the motion after effect illusion, and there's the troxa fading for the experts. It's really a very, very beautiful one because it makes this point so obvious that here you see what's not, that color is really a construct of your brain, a complete construct of your brain, based on lawful property. It's not random, it's lawful, and it does exploit something about the physics of the world, but it's really constructed, it's not out there. Do any of you dream in black and white, by the way? I'm always curious to ask. You only dream in black and white? No. Your brain is the older model.
Analog and digital.
So you all dream by and large in color, right? Because it's a question. And for example, a question I've always asked, do you have after effects in your dreams? I was wondering how to test that. You should expect after effects in your dreams. Hasn't been tested yet. All right. So in the color, so the third principle, so that's opponency, the fact that colors are represented, seem to be represented in opposite way, green minus red, blue minus yellow, white versus black. Trichromacy is the first one. The third one is color constancy, so that color of objects look more or less the same. So here, for example, it's a picture I just grabbed of the web. You see a balloon, deflating slowly on the ground, and this is the same color, and this is, of course, the same color. Now, they're lit very differently because this is sort of still billowing out. The sunlight only strikes this surface. This is in the dark. But by and large, you tend to see, I mean, you can notice, of course, there is a difference, right? If I ask you to, they're clearly not the same, but the color you tend to assign automatically, this is the same, and this is the same, and this is the same. And people do a lot of work on this property of color constancy. So here's another case. So this is the author, Young, who photographs. So he has these palettes of color, and just focus on this pink here. They look very different, right? So here you can see, so here it's the same image, just seen through a pink lens, filtered through a pink lens, right? The skin looks very different. But so the underlying surface is actually the same, right? It's exactly the same if you just focus on the surface. But in this case, they actually do look different, although the surface is the same. And here's a very famous color constancy illusion by Ted Adelson at MIT. So A and B clearly are different squares, right? A is dark square and B is a bright square. Yet, of course, I wouldn't be telling you this. Right? They're, in fact, exactly the same brightness because that's the way the shadow, it's playing around with the shadow in very clever ways. And there are endless amount of illusions like this on the web. The same color here, but different way here. So that has to interpret. So color constancy can be overridden by other cues. So here the Q is shadow, and our brain automatically assumes the shadows here to the left. We automatically know the light source. Our brain assumes our light source is up here on the right. Most painting, it's almost always on the web. And if you look at embossed fonts on the web, it's almost always embossed in such a way that the implicit assumption is that the light comes from the left. And we here the light comes from the right. And so we observe these patterns and, of course, immediately infer, well, this has to be brighter than this. And so our brain overrides. So it's a very flexible, so once again, surface is a very flexible computation that's a function of all sorts of other properties, not only wavelength reflection. But in this case, it includes knowledge of this shadow, which is not a local property anymore, right? Because in order to evaluate the color of this pixel here, for instance, the brain has to have some knowledge of the existence of the light source, and there's a physical object between the light source and this surface that casts a shadow. So it's a very complicated, it can be a very complicated computation.
If the green blocking object is not there, but only a shadow projected as if we're somewhere.
It becomes weaker. In fact, there's a very beautiful illusion that removes that exactly does that away. So I mean, in vision, as I mentioned before, none of these cues are absolutely essential, but the more cues they add, the more powerful these effects become. All right, so this is, if we now move, so this is physics and psychophysics, the way we experience things. If we now move into the nervous system, so the first stage that's relevant is where physics meets the brain, which is at the level of the photoreceptors. And most of us, I mean, I bet you there must be a few colorblind people here. One? There must be, statistically it's unlikely. There must be more here. You're unwilling to admit it. Anyhow, so here you can see this is the short wavelength photoreceptor, peaks at 420. For most of us, then 534, the medium, and then the long at 564. And this is the cone photoreceptor, the rod photoreceptor spectral sensitivity, which lies somewhere in between. Now, in fact, it's claimed like at dusk or dawn, when the light, there might be rare conditions under which you can use, so typically, like under these light conditions, the rod system, of course, there are many more rod photoreceptors than cones. There are roughly 100 million rods in us, and 3-4 million cones. But these typically in the lights like this, too bright, they're saturated. They're a rare condition like at dusk or dawn when there is enough light that these systems, the cone is still active, but that the rods are also active, under which you might see enhanced colors. Because then you would have, under those conditions, you would have 4 photoreceptors. Now, as I mentioned, there's these allele, and there are 2 different types of L photopigment that they differ by a couple of nanometers. What's amazing to me that here you have a single nucleotide polymorphism that you can, the effect of this shows up very discreetly in visual psychophysics. You can measure it. And as I mentioned before, if one woman in her eyes has in the different photoreceptor expressed both alleles, then she would have, the 2 L1 and then one M and one S. So she would have 4 cone pigments. The principle of univariance says that once a photon that has a particular wavelength, i.e. that has a particular energy, is absorbed, then the response of the nervous system to that photoreceptor will be the same no matter the wavelength of the incident light. So in that sense, all wavelength information is lost, and in that sense there are no blue photons. In a sense, what I mean by that if you get, so if you look at this, what many people would call a blue photoreceptor, well, for example, it also has a certain probability of a photon over here that's sort of more shifted towards the green part of the spectrum being absorbed. And once it gets absorbed, once there's a photoabsorption here, and this S photoreceptor signals, then of course the brain doesn't know, well, was it at this wavelength, or was it at this wavelength, or this, or this? It's just most likely to be here, but you don't know. And the only way the brain can extract color information, wavelength-specific information, is by comparing by comparing at least 2 of them. So that's why most animals, including mice, have two different cones, and some animals, including some monkeys and all apes and all humans, have 3 cones. So that's the principle of univariance. Once an absorption takes place, there's no more wavelength-specific information. All you know that S has signaled so many events, and M and L have signaled so many events, and now the brain has to compute with these relative energies. Yeah, so once again, that implies that two objects, this again is the Metamer, with the same receptor activation will look the same. So you have two different objects that give rise to the same activation in the S, L, and M cones, then they will look the same. All right, so in invertebrates, you can see here, so these just shows you the variety. So this is us. There's a rhodopsin here, roughly at 500. Then there's our sorry, this is actually, yeah, this should be the S cone, the M cone, and the L cone. Now, other animals like chicken, they have more, and there's some animals like shrimp that may have up to 12 cones. So once again, there's nothing universal about 3. It's not a principle of physics that we have to have 3 cone photoreceptor. It's something about our particular environment in which our species evolved. It's a very important point to make. And mice evolving differently, occupying a different ecological niche, they have 2, green and blue, that shifted towards 360 or 365 here. Quite, almost 100 nanometers shifted into the UV. Now what's interesting, Clay and I have had many debates because I was sort of shocked. I was actually shocked when I realized last year that almost every single paper that's been published on mouse vision, which we're now contributing, only uses green stimuli. Right, so that's trying to explore the visual system by just using, green things. I mean, you could do it, but you definitely may be missing a lot of the picture, and I'll come back to that. If you don't ever use stimuli, this is a little bit more non-trivial because we don't have, normal. If you buy just a normal CRT or something like that, it doesn't emit here because we are not sensitive to that. Right, so you have to build special apparatuses, which can be done and has been done by J9C here at UW, but you have to go to special lengths to stimulate it, which is why almost everybody works using this, using the green cone. Now, this is a remarkable picture. It's a picture of 2 living human retina of 2 individuals. And what you can see is 2 really astounding, this came out in Nature a while ago by Williams at Rochester, and really quite astounding because it shows you, first of all, so this is a little parafovea. It's outside, a couple of visual degrees outside the fovea. Fovea is here, point as sharp as seeing, so it's somewhere here. And they actually visualized these living retina. He constructed a special sort of inverse scope, if you want, to look at the photoreceptor, and then they're artificially colored so you can see the 3 different types. So a number of things to observe. First of all, and this was known before, in both individuals, there are very few S cones. This tends to be typical. So unlike a camera, right? So if you look at the same thing on a camera, what you'll see hexagonal spacing throughout the entire camera, just like here. And the fovea, it's very hexagonal spaced. But you see a very regular arrangement of 3 different, it's done using silicon circuit technology, and it's done that you have 3 very regular spacing of the 3 different photoreceptors with 3 different spectral sensitivity, and they're all exactly equal. Well, in the eye, that's not true. Typically, we have between 5 and 10% of our cones are S. That's why you should not use text on slides in PowerPoint presentation, use blue text. Because people, by and large, and you can see this resolution we have in the blue is much reduced compared to the resolution in the green or in the red. Partly that's just we have many fewer S cones. All right, then here we can have the other point I want to make that can be quite different. So in this case, 3 out of 4 photoreceptors are M. In this guy, there are only 2, 1 every second is, so there can be quite a difference amongst people. And furthermore, even within a single retina, this is nothing like the regularity of a nice silicon camera, your CCD or CMOS camera, where everything is perfectly regular. It's quite irregular. And it's really a wonderful job. It's sort of the general job of perception. It's sort of a con job partly to suggest that everything is beautiful and regular. But in fact, if you look at our very receptor equipment that's already responsible for sampling the visual environment, it's already very irregular and quite noisy. It's still, the world doesn't look like that. If I look at a red surface, or if I look at complicated properties, it doesn't look this irregular, and that's the job of the brain to partially compensate for all this irregularity. So it's really quite interesting. So it differs a lot among photoreceptors. There's a lot of variability across people, and within the retina you can find patches that are primarily L dominated, and you can find other patches that are primarily M dominated, which would suggest that if you do very careful vision, very careful with stabilized eyes, you should see those differential spots where you're very sensitive to L or very sensitive to M. Now notice, so this is a 5 arc minute, so there's 60 arc minute in a degree, and typically your thumb at arm's length is 1.5 degrees. So these are very small distances. So typically your eyes, because it moves constantly, right, it has these jitters, and then of course it makes rapid saccades. So typically you would smooth over that and integrate that. But if you do careful psychophysics, it should show up. All right, so there are 3 cones. In most of us, they're not distributed evenly. And color arises out of comparing the response among these receptors. So what the brain has to do, it has to compare among L, M, and S cones in order to extract color. There's nothing special about 3 cone types. That's just what we evolved with. Other species have fewer, like most mammals, like dogs and cats and mice only have 2. And some species have many more than 3, up to 12. And even with humans, there's variability. And this was discovered. This remarkable fact was only discovered by Dalton in 1805 or something like that. And thus, in some old books, it's still known as Daltonism. The fact that there are some of us, and because it's X gene linked, as Clay said, it's particularly the men that are not truly colorblind, although they're called colorblind, but they have, well, they can have anomalous 3 cones, or they can only have 2 cones, or rare 1 cone, or even more rare where they have no cones, so-called achromats. So you will know this because, all normal trichromats, these are a series of color plates that were developed by a Japanese doctor, Ishihara, in the early part of last century, that can be widely used, for example, in the army and in schools to test for the existence of of various forms of colorblindness. And depending which exact form of colorblindness you can have, you can test, because there are different sorts of colorblindness depending exactly on which mutation you have. So you should all be able to see 25. You can see 25, Mike? And what's that number?
I don't see anything in this.
42. And that?
I don't see anything in there.
45, and that? 8. So I'm sure there's at least one or statistically more of you that should have difficulty with some of these colors. So those are very easy ways. You can just buy them and have one in the lab. You can just use it. So if you're a particular type of colorblind, a red-green deuteranon, nope. Well, then you're badly messed up. So this is what the world looks to some type of colorblindness. As I said, there are different forms of colorblindness. And so one difficulty people have of this sort of colorblindness is that they can't tell whether their skin is burned or sunburned. They can't tell the gorgeous color of this Apple Macintosh. And also traffic lights sometimes. That's why in some countries they have vertical traffic lights, not horizontally anymore. But they have difficulty distinguishing between red and yellow warning lights. So, I mean, it does look quite different, right? And what I find remarkable that until roughly 200 years ago, this fact wasn't appreciated, that roughly 1 out of 14 men has this colorblindness, and that we don't see the world in the same way. Statistically, it's roughly the same, but we each have slightly different perception. And if you remember that patch I showed you of Williams, every one of us sees literally the world slightly different, because we have a different spacing and different distribution, different densities of L, M, and S cones. The vast majority is X-linked, so it's roughly 7% in males and under 0.5% in females. So then there's this very fascinating subject of achromacy. And if you want to find out about it, there's a movie and a wonderful book by Oliver Sacks, my favorite neurologist. It's called The Island of the Colorblind, where he travels with a Norwegian psychologist called Hansen, who's actually colorblind, who grew up achromat, although he later on became an expert in vision, and travels to this island in the Marshall Islands that was visited by a typhoon roughly 250 years ago that wiped out a large fraction of the population except the king and his harem, all his ladies. And the king was presumably carrier of this anomaly And because a significant fraction, like 20% or so of the population today on this island, doesn't see color at all. Now they have all sorts of other deficits. They have to wear very, very heavy sunglasses. They have a nystagmus. But one thing, they can't see color. And Oliver Sacks visits them, and he has a BBC crew following them around. If you want to Google it, it's really fascinating. Because it addresses an old question that's been asked in the philosophy of mind literature is, what happens if you knew everything about color? You knew all the theory, you know, in principle, apple is red, and you know the American flag is red stripes and all of that, but you just didn't have the genes for color. Would you still see color? And the clear answer to my mind is empirically no. If you don't have those types of receptors, no matter how hard you try, no matter how much you know about color, and you can also see this in this visual psychologist, the person who accompanies Oliver Sacks, he don't see color. It's something direct biological. It's not an inferred property. It's not a property that you have to sort of psychologically, mentally, consciously infer. You perceive it. Although the brain, as I try to show, has to do a lot of computation to actually extract it. Anyhow, very beautiful story. Now, there's an interesting story here also why most mammals have two types of cones. And an S1, we all have an S1, and then just like the mice, another one that sort of is somewhere between green and between L and M. And some primates, some New World monkeys only have two cones, while all Old World monkeys, like macaque monkeys, have three cones, and some New World monkeys have three cones. In particular, I'll show you an experiment in a second. So this is the world if you just have intensity. That's the world if you have two cones, and this is the world if you have three cones. Now observe the fruits and how much more you can observe about fruits and how ripe they are when you compare two cones versus three cones vision.
So you said cats and dogs have two?
Yeah. In fact, don't all mammals have, except some primates, have two cones? Yeah. So there are these squirrel monkeys that live in South America, where most males, or some fraction of males, have two cones, and all the females have three cones. And it's interesting, here they eat fruits, and the troops are led by female on their search for fruits. Now there's an experiment done here by the Jay Nines and his group here at UW, and he gave a talk. I don't know whether you all attended half a year ago here at the Institute. What he did, he cured, quote, male dichromat squirrel monkeys of their dichromacy by introducing in an AAV virus and directly injecting it into the eye, a couple of trillions of particles of virus, the L photopigment. It's a pretty cool experiment. So 6 months later, he could see both in the retina using reporter GFP, as well as doing behavior, more interesting, because, okay, so there are two questions. First of all, does this technique work? And he showed, yes, photoreceptors, some fraction of the M photoreceptors do begin to express the L photopigment, the one that's introduced through the human gene in the form of the AAV virus. Now, more relevant question is, does the brain take it? These are, after all, now already adult, I think these are already adult monkeys. They grew up with dichromacy, and so will their brain have enough plasticity to make use of that information? And the clear answer is that a behavioral test where you can do sort of essentially these matching tests that I explained, where the monkey could only do it correctly if he had access to the third photopigment, that they actually take advantage of this, which is quite remarkable. What that means, and if you talk to him, he feels that, you know, once this can be done routinely and it's done safe and reliable, and of course AAV has been approved for certain types of gene therapy, this is an operation that should be routine and just like LASIK. It should be an operation, you go to your ophthalmologist, he injects a virus, and then a couple of months later, who knows how long it is, you can see color. So we'll see how long it actually takes to go into clinical practice. But given that there's 7% of guys who don't have color, that is potentially a very large number of patients who could be, quote, treated in this way. I put treated because it's unclear whether this is really, you know, a pathology. It would help him, you know, it would help him to enjoy literally larger spectrum of life.
Would that pass through to progeny, or would that stop with that?
No, because it wouldn't be in their sex line. No. So this is here. This is also from ninth. So this is what I mentioned before. This is now in mice, in fact C57, our type of mice. The spectral sensitivity measured in the retina, and you can see there are two cones here. So these I just added. These are the human peak sensitivity of our S, M, and L cones. And this is the M cone, and this is the S cone. So the S cone is shifted roughly from here all the way over here. Now look at the magnitude here of this compared to this. Much bigger. This is the UV part. And they also did some behavioral tests where the mice had to do some very simple behavior. And again, the response was much stronger in the S portion than in the M portion. But all the vision that certainly we're right now doing at the institute, or that everybody else is doing out there, with two exceptions, two groups that probe UV, is always done here. Now mice have another complication. This is data that was taken by, was work of a surf student, summer undergraduate student from MIT, who worked with Stefan and Vihalas over the summer here at the institute. So she took data from Jay Nines's lab. So this is so the mouse retina. It's roughly factor 10 smaller. It's 2.5 millimeter versus 22 millimeters or 25 millimeters in the human eye. And she expressed here the gradients because so we have photoreceptors to first extent mainly in the cones are mainly in the fovea, and then they fall off. Mice have something very different. They have this gradient that goes from top to the bottom. They don't have a fovea. They have sort of more these elongated. Their eyes are much more lateral than our eyes, that are forward placed. And so what they have this weird gradient where in the upper part of the retina they have M, in the lower part of the retina they have S. Now, of course, the lower part of the retina looks at the sky, and this one looks at the ground. And so that's one peculiarity, that they have this grading that at any one location you tend to have more one or the other, although somewhere here in the middle they equilibrate. So that's very different from us. The other peculiarity is that actually well, there are several systems, but a lot of the cones co-express both the S and the M pigment, which is really strange. It's very different. Right? We either have we have a cone that either expresses the M or the S or the L opsin, and in mice, a lot of the cones co-express both. So it's
Is there UV sensitivity likely to that same ecological niche?
well, okay. So why do they? You can ask why do they have UV? And we had another student, Caltech, a surf undergrad student, working with Vilas and Ram and Stefan over the summer, looking at exactly that question and looking at UV scattering. So they bought the two cameras. They have a UV camera that has exactly the same spectrum, which Stefan has exactly the same spectrum as a mouse, and a green spectrum, the M spectrum, and then compare. So I think our feeling is that one of the biggest predators of mice are birds of prey like owls. And so I told this story before when I went climbing up at Vantage at night there, you can see at midnight under the moon, you see these mice running around very quickly. They're so amazingly fast. And there are also owls there, and they get beaten by owls. And so if you look at the spectrum, in particular after the sun has set or before the sun has risen, because of the Rayleigh scattering, which is 1 over lambda force to the force power inverse to the force power of the wavelength, that due to that scattering, the reason why the sky is blue is that you would observe the UV for long after the sun has already set. So it's already dark, but there's still UV photons around. And same thing, it may be dark in the morning, still dark, but there's already UV light around. So that might be sufficient for them to detect whether there's a predator flying overhead. And it's interestingly that these guys are predominantly in the lower part of the retina, which is mainly sensitive to light from above. So there may well be a bunch of specialized systems, or at least one specialized system that's observed this, about which right now we know nothing. All right, so to go a little bit more into the retina. So, these information from these different cones, M, L, and S cones, is pooled in complicated ways that people are now beginning to understand. At the output of the retina, you really have what's known as a color opponency. So once again, remember that psychologists for the longest debated whether the brain or the mind uses trichromacy to represent colors or uses opponency. And the clear answer is yes. Like in some of these, like the wave particle dispute. It actually does both, depending on where you look in the nervous system. So the output of the retina, the information that gets shipped on the action potentials onto the rest of the brain, here is already coded in an opponent way, such as that you have particular types of ganglion cells. In the monkey, they're known as power-cellular cells and some coniocellular cells, where you have, for example, so this would respond, I'll show you one, this would respond to onset of red light and gets inhibited by removing red light. And this gets inhibited in the surround. So it's both, so spatially it's an opponency center surround, just like Clay Reed showed you in an earlier lecture. But then the center and surround have different wavelength sensitivity. So this mainly responds, this gets excited by red light and gets inhibited by greenish light. This cell is the opposite. This cell gets inhibited by red light in the center and gets excited by green light, and here you have the exact opposite. So here you have a cell that gets excited in the center by green light, and vice versa. So a lot is known about the green-red channel because we have a lot of cells that and a lot of cones that subserve this channel. Then there's a second, there's another channel. Remember the Hering's opponency principle. There's channel that carries intensity information. Every time we watch a black and white movie or black and white TV, that's the only one we use. And then there's both red, green, and blue, yellow. So this is a blue-yellow representation where in fact they know what the ganglion cells look like. And here you have an on response in the center to blue light and an off response to yellow. Yellow being a combination of red and green. And these are known as so in the monkey they're known. This is a ganglion cell, so-called coniocellular ganglion cells that project to the coniocellular layers in the LGN and then onto cortex. So Clay has been active in this area, one of the many areas in the visual system the monkey he's been active in. He had an earlier Nature paper, I believe, on this topic, and this is a later Neuroscience one, where Clay and Bob Shapley looked at, this is in the monkey. This is in the LGN, so halfway between the retina and the primary visual cortex, and where they look at two different types of cells. So these are so-called magnocellular cell, and this is a powercellular cell. So in the monkey, you have this nice layering of cells that have differential size. So they're called big cells and small impoverished cell. That's just Greek or Latin for big cell and impoverished cells. And these cells, by and large, magnocellular cells, they don't carry wavelength information. So here you can see, so let me see. This is the on center, so blue is off. Okay, so here when you use L isolating stimuli, so this tends to be more reddish stimuli, and you shine them in the center, the cell here is turned off and gets excited by red in the surround, and sort of here is the same for sort of greenish looking like stimuli that maximally activates the M cone. And so if you look at the luminance channel, which combines across the different cone types, what you see is sort of a center surround organization that doesn't carry much color wavelength specific information. So by and large, they don't carry a lot of wavelength specific information. The ones that seem to carry the wavelength specific information are these powercellular cells. So here's one type of cell. So again, they have a center surround structure, just like in the retina. In fact, not much difference between retina and LGN. And they get excited here. So this cell gets excited by sort of reddish stimuli in the center. And this cell gets inhibited by greenish stimuli. Blue means off. Gets inhibited, you can see, by greenish light in the center, and so you have a typical on-off organization similar to the one in the retina. So between retina and LGN here, there doesn't seem to be a lot of change, and these are called, once again, these are the opponency cells. Now in the cortex, you get to a more complicated structure. You get double opponency. You get cells that seem to represent, for example, red in the center, but then they also get inhibited by green in the center, and they might be excited by green in the periphery, and they get inhibited by red in the periphery, right? So most likely they have to combine the output of some of these simple opponency to get rise to double opponency. But you also see some more remarkable observations. So it's known since many years that if you look at tangential view of the cortex, you get these blobs of cytochrome oxidase. If you stain in particular for an enzyme involved in energy metabolism, you can see that it's not in layer 2, 3, it's not homogeneous, but you have these blobs. And it's known since David Hubel days, David Hubel and Wiesel and Marge Livingston's day, that these are chock full of wavelength specific, of cells that signal wavelengths. It's not by and large in the interstitial spaces between the blobs, it's really in the blobs. And this is a paper by Chatterjee, it's going to be here at the Institute in a month's time, I think. And Clay, I think this is still unpublished, where they look at detail using 2-photon calcium imaging, once again in the monkey, where they look at inside, the structure inside, here's one blob and here's another blob. And what they can see, there's really very nicely, he can see the different cone isolating stimuli. So this tends to be red-greenish, here's sort of more bluish-yellow. What you can see there in this blob, so this is 100 micrometer, and here they focus in on this one blob. You can see there's nice spatial structure that different wavelengths, specific information, cells are represented differentially. Right, so here it tends to be more bluish, here's more greenish, here's more sort of yellowish in the center. So there's this nice spatial organization in the architecture that you can see. So this is what the way people think of it in V1 in a monkey. You have this a column in the monkey with these different layers, and you, you have specific information that can send corneal cell that's blue-yellow. Here tend to be red-green or green-red cell that combine here and then combine to give rise to more sophisticated, I'm almost done, color computation in these blobs. It's the last slide, or last two slides. We know very little about representation of color beyond V1, although there's quite a bit of fMRI. Some of it's controversial. There's a very nice, Jack Gallant, some of you may know, published a study in Neuron a couple of years ago with a remarkable case of a single patient who had what's called achromatopsia. So achromatopsia is not color blindness conventionally. So typically these are individuals that see color normally, and then they have a stroke, and then they don't see color anymore. The world becomes strangely denuded of color. The most famous one is probably described in Oliver Sacks. He described a painter who had a stroke and who recovered from the stroke and couldn't really paint in color anymore. Here you have an individual who was basically, after some transient loss, was fine, but what they found, to his own surprise, i.e. to the patient's own surprise, that he was colorblind in the upper left quadrant. So he saw color normally here, here, and here, and he had some form difficulty, but more or less a pure color difficulty just in his upper quadrants. Very specific, only to here. He himself never noticed this. You know, there are always weird things going on in your visual system, at least in mine, and you know, you don't necessarily pay attention to them, but it's very reproducible, and it's due to a very small pea-sized lesion in the ventral part of V4. So it's a beautiful structure-function link, right? Because it says if you, at least in this guy, if you lack neurons in this part of the brain, you'll get a very specific loss in this specific computation of color. It's nothing to do with retina, right? The retina is perfectly normal in these individuals. In fact, their color was normal until they had this stroke. All right, so in summary, color is an attribute of surfaces that we are conscious of and that we use in our everyday vision. Color has no direct correspondence to a single physical quantity. It's a compared quantity among, in most of us, among three neuronal population, and depends on all sorts of other things. It depends on multiple types of wavelength sensitive photoreceptors. How many photoreceptors are used varies across individuals, i.e. from humans from 0 to 4, and across species from 2 to 12. And color is then subsequently represented in an opponency manner and is computed, at least in us, in higher order visual cortex. We know almost nothing about to what extent color is exploited, given these curious facts about the visual system of the mouse, to what extent is exploited and represented in higher order visual cortex in mice. And with that, I thank you for your attention. Oh no, people have done a lot of lots of psychophysics. In fact, it's been actually not easy to find. So for example, object naming, object identification, you can do black and white just as fine. People have done lots of experiments there. So it's actually not quite clear when you remove color from images, unless you make them, when you remove color from images, what the big loss is. It's remarkably unclear given the fact that we all love color. Given the choice, we instantly prefer a color television or color monitor over a black and white one. No, so there's quite a bit of psychophysics. There has not been a lot of physiology because actually it's not very easy to keep things because of color constancy. The fact that, for example, depending the exact environment, the light environment of your lab, that really influences color. You have to very careful control for all of that, and that's not easy. And there are not all that many investigators compared, for example, to the number of investigators who investigate stereo or motion. Yeah, the question was whether in principle, rather than introducing a virus for, let's say, the missing photopigment, whether you can introduce something artificially and build something that would be sensitive to UV or to infrared. Yes, and I think it's entirely possible. What these experiments suggest, at least in monkeys, that there's enough plasticity, there's enough reservoir of plasticity even in adult monkeys that they can adapt to that. And that's presumably also true, although the younger you would get this operation, the better it is. Yeah, and I think there are going to be some cool, interesting experiments people will can do. Of course, we can always just buy an infrared, or we use an infrared sight, right? That's much easier rather than engineering and waiting for half a year. Plus, it's not very invertible once you have it. But yes, it's possible. All right, thank you very much.